Antibody structure. An antibody is composed of two heavy chains, 50 kD each, and two light chains, 25 kD each, which are joined by disulfide bonds to form a Y-shaped structure, 150 kD. Antibodies are further divided into two regions, a variable region and a constant region. The variable region is responsible for the antigenic specificity of an antibody. This region includes a fragment antigen-binding, Fab, portion that binds the antigen with high specificity. There are two Fab portions in each antibody, which can simultaneously bind two identical epitopes, a specific antibody binding site of an antigen, of a particular antigen. The constant region of an antibody includes a fragment crystallization, Fc, portion that binds cell surface receptors, Fc receptors, on circulating WBCs, macrophages, and natural killer cells. This binding is necessary to initiate an immune reaction. In addition, there are two hinge regions that join the Fab and Fc portions of an antibody. What are the types of antibodies? IgG. This isoform accounts for 70 to 75% of all human immunoglobulins found in the blood. Depending on the size of the hinge region, the position of disulfide bonds, and the molecular weight of the antibody, IgG can be further divided into four subclasses: IgG1, IgG2, IgG3, and IgG4. In general, proteins are responsible for triggering IgG1 and IgG3 production, whereas IgG2 and IgG4 typically respond to foreign polysaccharides. IgG is the main component of the humoral immune system, immune response initiated by macromolecules present in the extracellular fluid, because of its abundance. Due to its small size, monomeric, and high diffusibility, IgG is the prevalent type in the extracellular fluid that binds Fc receptors on phagocytic or other lytic cells and initiates the antibody-dependent cell-mediated cytotoxicity, ADCC, response, a cell-mediated defense mechanism wherein effector cells, phagocytes, destroy the target cell. In addition, IgG triggers phagocytosis to initiate opsonization reaction, a process used to destroy foreign particles, e.g. bacteria, through phagocytosis. Apart from these functions, IgG is the only antibody that can cross the placenta and provides passive immunity to the fetus and infants in the first few months of life, which facilitates NK cell responses and induces the production of interferon-stimulated genes, ISGs, and to antigen presentation and/or stimulation of B and T cells, see text. HIV-1 infection impairs diversification of an IgG antibody response to downstream isotypes. RIIA. Activation of PDCS leads to the production of IFN-RIIA, particularly in individuals carrying the 158V genotype. However, it is proposed that multimeric IgG2 antibodies primarily broaden the function of the antibody response by enhancing phagocytic activity against GAG proteins associated with HIV-1 RNA, as a consequence of the functional characteristics of IgG2, see Table 1, which activates plasmacytoid dendritic cells, PDCS, via FcRIIA. Upstream IgG isotypes, IgG2 and possibly IgG4, may also contribute to NK cell activation by ligating Fc. A diagrammatic representation of how isotype diversification of IgG antibodies against HIV-1 GAG proteins might enhance antiviral accessory cell responses against HIV-1 infection. It is proposed that IgG antibodies bind to HIV-1 GAG proteins expressed on the surface of cells infected by HIV-1, including resting CD4 plus T cells. 9. Activation of natural killer, NK, cells is elicited by downstream IgG isotypes, IgG3 and IgG1, via Fc IgM. IgM is the largest antibody and the first one to be synthesized in response to an antigen or microbe, accounting for 5% of all immunoglobulins present in the blood. IgM typically exists as polymers of identical subunits, with a pentameric form as the prevalent one. In its pentameric form, 5 basic antibody units are attached by disulfide bonds. Other forms include secretory IgM, which is synthesized by glandular-associated B cells, and monomeric form, which is present in the B cell membrane and functions as a B cell antigen receptor. Due to its large size, IgM is mostly intravascular and has a lower affinity for antigens. However, since pentameric IgM has 10 antigen binding sites, it has higher avidity, overall binding strength, for antigens than IgG and acts as an excellent activator of the complement system and agglutination. IgA. It accounts for 10 to 15% of all immunoglobulins and is prevalent in serum, nasal mucus, saliva, breast milk, and intestinal fluid. It has two subtypes, namely IgA1 and IgA2, which mainly differ in terms of their hinge region characteristics. At mucosal surfaces, IgA provides the primary defense against inhaled and ingested pathogens. The vast surfaces of the gastrointestinal, respiratory, and genitourinary tracts represent major sites of potential attack by invading microorganisms. Immunoglobulin A, IgA, as the principal antibody class in the secretions that bathe these mucosal surfaces, acts as an important first line of defense. IgA, also an important serum immunoglobulin, mediates a variety of protective functions through interaction with specific receptors and immune mediators. The importance of such protection is underlined by the fact that certain pathogens have evolved mechanisms to compromise IgA-mediated defense, providing an opportunity for more effective invasion. IgA function may also be perturbed in certain disease states, some of which are characterized by deposition of IgA in specific tissues. This review details current understanding of the roles played by IgA in both health and disease. IgE. IgE is the least prevalent one, with a serum concentration 10,000 times lower than IgG. However, the concentration of IgE increases significantly in allergic conditions, such as bronchopulmonary aspergillosis, and parasitic diseases, such as schistosomiasis. In response to pathogens, IgE binds to mast cells via specific receptors, followed by pathogen-mediated cross-linking of these receptors, degranulation. This causes recruitment of eosinophil at the site of infection and destruction of pathogens via ADCC-type mechanisms. It primarily provides protection against helminth parasites but can also respond to foreign substances even in small amounts and is designated as a gatekeeper. Allergic reactions are predominantly associated with IgE. The function of IgE antibody as mediators in allergic reactions of type I is explained by their ability to interact both with antigen and with receptor molecules on the membrane of blood basophils and tissue mast cells in the human body. Antigen-induced cross-linkage of receptor-bound IgE initiates a process that results in the release of histamine and heparin, which increase vascular permeability and promote contraction of smooth muscle. IgE-mediated mechanisms are conventionally known to facilitate degranulation of mast cells and basophils and promote Th2 immunity, mechanisms that are not only directed to mounting an appropriate defense against parasitic worms, noxious substances, toxins, venoms, and environmental irritants but that also trigger vigorous allergic reactions in patients having allergies. IgE has an essential role in type I hypersensitivity, which manifests in assorted allergic diseases, such as food allergies, allergic asthma, most types of sinusitis, allergic rhinitis, and specific types of atopic dermatitis and chronic urticaria. In certain cases or conditions, high IgE levels indicate that the body is overreacting to allergens leading to an allergic reaction. It can be considered as a sign that the body is fighting off an infection from a parasite or with some immune system conditions. In addition to a role in allergic reactions, IgE plays a significant role in the immune response to parasites such as Schistosoma mansoni, Trichinella spiralis, and Facilla hepatica. IgE is utilized during immune defense against certain protozoan parasites such as Plasmodium falciparum. Release of histamine induces muscular contractions in the intestine, which contribute to the expulsion of parasites. IgE also appears to play a role in resisting ticks of the Ixodidae family, which carry among other diseases the organisms for Lyme disease and ehrlichiosis. However, the exact workings of IgE remain unclear. IgE also plays a significant role in responses to allergens, such as anaphylactic drugs, bee stings, and antigen preparations used in desensitization immunotherapy. IgD. IgD functions as a B-cell antigen receptor and may participate in B-cell maturation, maintenance, activation, and silencing. Although the exact function is still unclear, IgD may be involved in humoral immune responses by regulating B-cell selection and homeostasis. The function of IgD has been a puzzle in immunology since its discovery in 1964. IgD is present in species from cartilaginous fish to human, with the possible exception of birds. This nearly ubiquitous appearance in species with an adaptive immune system demonstrates that IgD may be as ancient as IgM and suggests that IgD has important immunological functions. In B cells, the function of IgD is to signal the B cells to be activated. By being activated, B cells are ready to take part in the defense of the body as part of the immune system. During B cell differentiation, IgM is the exclusive isotype expressed by immature B cells. IgD starts to be expressed when the B cell exits the bone marrow to populate peripheral lymphoid tissues. When a B cell reaches its mature state, it co-expresses both IgM and IgD. A 2016 study by Ubelhart and colleagues found that IgD signaling is only triggered by repetitive multivalent immunogens, while IgM can be triggered either by soluble monomeric or by multivalent immunogens. C. Delta knockout mice, mice that have been genetically altered so that they do not produce IgD, have no major B cell intrinsic defects. IgD may have some role in allergic reactions. Recently, IgD was found to bind to basophils and mast cells and activate these cells to produce antimicrobial factors to participate in respiratory immune defense in humans. It also stimulates basophils to release B cell homeostatic factors. This is consistent with the reduction in the number of peripheral B cells, reduced serum IgE level and defective primary IgG1 response in IgD knockout mice. Camelid antibodies. Non-canonical immunoglobulins in camelid species. Camelids, alpaca, llama, camel, vicuna, and guanaco, produce two non-canonical immunoglobulins alongside the conventional IgG1. IgG2 and IgG3 are known as heavy chain only, or heavy chain, antibodies. They have single domain binding sites, VHH, and do not contain CH1 domains or light chains, figure 1. Jackson Immunoresearch offers a range of secondary antibodies utilizing the properties of camelid immunoglobulins, for detection in assays and to expedite VHH development. We also provide camelid-derived secondaries. Heavy chain-only antibodies. VHH antibodies, nanobodies, are rapidly being adopted in a variety of fields. Uses for these novel antibodies include various applications such as medical diagnostics, therapeutics, biosensors, crystallization partners, and detection of small molecules. VHH antibodies are derived from the VHH domain of heavy chain-only immunoglobulins, IgG subclasses 2 and 3, from camelid species. VHH libraries derived from a camelid host animal, typically alpaca or llama, are screened for target specificity, and the desired VHH sequences are obtained. Recombinant VHH, rVHH, can then be produced in an expression system of choice. The long CDRs shown in the ribbon diagram, figure 1, are characteristic of the VHH domain, and provide the antibody access to recessed antigenic sites not available to canonical antibodies. Benefits of VHH antibodies. Framework. The 12 to 15 kDa antigen binding VHH domain acts as a framework for recombinant antibody production. VHH antibodies can be designed in a variety of structural formats. Targeting. The small size and long CDR3 loop of the VHH antibodies allows better access to cryptic epitopes compared to conventional antibodies. Stability. VHH antibodies have good solubility and are stable to heat, proteases and pH extremes. Therapeutic advantages. VHH antibodies can cross the blood-brain barrier and access other remote locations, making them useful for drug delivery. They also clear quickly from circulation and are relatively non-immunogenic to humans, Banas et al., 2017. Good stability allows delivery via a variety of routes, including intravenous and subcutaneous injection, nasal inhalation and oral ingestion. Production. VHH antibodies are easy to clone and modify. They can be produced recombinantly at high levels in bacterial, yeast, and mammalian expression systems. Their design can include tags for purification, or functional groups to enable the addition of drugs or fluorescent probes.